afternoon, everybody. First of all, I would like to thank the organizer, CEFIPRA, the DST, and my fellow colleagues who have given me the opportunity to exchange some of our thoughts regarding the stresses which we are experiencing in our sales are experiencing in the micro channels. Okay, so mostly when we talk about stress, so by default it comes to our mind that okay, so since the cells they cannot be considered to be in an isolation state, so mostly they are in a biological fluid or a cellular media, so they will be experiencing the stress derived from the shear that is the fluidic stress. Now, other than fluidic stress, sometimes or often the cells they are also experiencing some of the thermal stress. Okay, so I will be expressing some of the thermal stress and how this thermal stress they are basically if we are having a regulated thermal stress, then perhaps how we can modulate the cellular viability. Okay, so the presentation outline is like so we'll be having a thermal stress strategy for the cells followed by cell isolation phenomena, then microfluidic bioreactor or a reactor in which one can say impose the thermal stresses, then the stress distribution in microchannel followed by some of the in vitro results. Okay, and here the stress when we are talking about the thermal stress particularly, in our cases we have considered the thermal stress to be the cryopreserved stress in a regulated fashion. Okay, so talking about the thermal stress strategy of the cells, so here you can see, as I told that here we are considering the thermal stress to be the cryo temperature derived stress. Okay, so the cryo temperature derived stress, if we are having say the isolated mesenchymal stem cells seated in say a Petri plate or on a seated Petri plate in absence of a shear stress. Okay, and then if we are going for providing or exposing it to a thermal stress of the cryo temperature. Okay, so there we can see that the cells they are mostly having a steroided microstructure. Okay, but here the stress, the cryo stress, it has been provided say instantaneously. Okay, so in that case we are going to get most of the steroided structure of the cells. Now, secondly, talking about the isolation of the cells, so it is a very well-known phenomenon of isolation of the cells. So in our case, we have isolated it from the umbilical cord. Okay, and then it is followed by proliferations and then sealing the cells in the microchannels. Okay, and then providing a regulated cryopreservation phenomena. Okay, so that is a uniqueness in our case where we have derived that. Okay, so if the cell is undergoing a cryopreservation say instantaneous cryopreservation, in that case the kind of viability the cells are showing, it is very much different when we are having a regulated imposition of a cryopreservation strategy. So this is a microfluidic bed bioreactor design. It has been shown over here. Okay, with different stages of blood forms in order to have both the cell culture as well as the thermal stress scope. At the same time, we can also have a scope view for a microscopic view. Okay, so here you can see some of it shows some results of the shear stress. Okay, so it is not like that we are having the say the fluidic chip with a static culture. We are having a fluidic chip with a dynamic culture, that is the dynamic flow of the media. Okay, and along in presence of the dynamic flow of the media, we are just having some microfilms within the channels, and that is providing some electrical current flow within the fins which will result in development of some of a regulated thermal stress over there. Okay, so here the numerical results, here we can see that when we are inducing some flow over there, it shows that near to the surface, that is near to the wall of the channel, we are having the stress is going to be maximum. Okay, along with the decrease in velocity over there. Okay, so the shear stress on the wall near to the inlet of these vessels, it can be seen to be a maximum. Okay, and within the channel, with increase in the depth of the channel, it is you can see with increase in depth of the channel, the stress is increasing due to the boundary layer phenomena present over there. Okay, so these are some of the governing equations which we have imposed while modeling the shear stress phenomena. So I am not going into this, as most of us are acquainted with this. Now in the in vitro studies, so here we can see it is showing a schematic of this magnifluidic device with the experimental setup. Okay. So here you can see that we are having, say, two syringe pump over there. So the syringe pump shows that here the fluid is not going to have a peristatic pulsatile flow. Okay, it will be having a linear flow. Okay, we had avoided the pulsatile flow in this particular experiment, as we are also say providing some regulated temperature infusion over there. So in order to just explicitly measure the effect of the regulated temperature, we have kept the flow to be constant. Okay, otherwise it is difficult to say separate out the effect of the regulated flow and the regulated temperature as well. Okay, so here it shows some of the results of the shear stress on the number of focal adhesion points, or on the recruitment of the focal adhesion points inside the channel. Okay, so in case of static control, what you can see, the focal adhesion points, it is somewhere here. Okay, and these focal adhesion points increases, number of focal adhesion points increases with the increase in the flow rate, and that is a very obvious phenomena, because the cells, they will always try to resist the deformation. Okay, which is happening due to the external forces, and that it can do by creating more number of focal adhesion points between the cell membrane and the substrate. Okay, now the interesting thing is that if we are performing this phenomena, say with a cryopreservation strategy, where the temperature, it has been, say, or the thermal shock, it has been provided without any kind of regulated thermal shock. Okay, so there you can see there is a rapid decrease in the focal adhesion points. Okay, now in order to say regain or to have a sustained focal adhesion points, what we have done that, okay, so let's go for a regulated cryopreservation. Okay. So this is a slide just showing that how we can quantify those focal adhesion points by quantifying the number of cytoskeleton tension recruited in the process of the cell, recruited in the process of the cell cryopreservation phenomena. Okay, so here you can see that the first step is that we need to do a image thresholding, where we'll be deriving with the histogram of the Gray level intensity. So obviously, if we go back to the previous slide, here you can see that we are having we are having some colorful images with the cytoskeleton as well as the nucleus and other protein factors expressing them to the fluorescence, obviously. Okay, now in order to measure the number of focal adhesion points, so number of focal adhesion points if we consider that the cell is responding in a linear fashion with respect to the external forces where the temperature is constant. Okay, so in that case, what we can see that the more the number of cytoskeleton tension is directly correlated with the number of focal adhesion points recruited at the cell membrane, or you can say at the transmembrane protein scale. Okay, so accordingly, so the idea will be to identify the number of cytoskeleton getting, say, in a striated form or in a tensile form, and then we can just map it in the form of number of focal adhesion points. So for that, first the image thresholding will be done. Okay, then it will be followed by the image segmentation. So in the thresholding, what we are going to get? So we are going to get a spectrum of the gray values. Okay, so in the form of histogram, and from there, what we are going to do? We are going to select what are the corresponding, say, histogram bar graph, which is corresponding to the cytoskeleton. Okay, and then we can do the segmentation of the image based on the threshold histogram profile. Okay, now once we have done the segmentation, so in the segmentation, so again we can divide the entire image into different regions, and on those different regions we can have the segmentation. Okay, like region 1, 2, 3, 4. So within the region 4, further say within any one arbitrary region we can have, say, further segmentation of in a quadrant form, and then within each individual quadrant we can have that. Okay, so the more number of, say, discretization of the spatial information we are going to perform, what we are going to derive out of it, that is the more precision in the information regarding the striated cytoskeleton, which will help us in having a much better quantified results of the focal adhesion points. Okay, so here it shows the effect of shear stress on the cell adhesions. Okay, so if we talk about in terms of the cell viability, okay, so in terms of cell viability, so in this figure you can see we are having the one graph it is showing, or one bar it is showing, so without shear stress, and the second one is with shear stress. Okay, so with shear stress in a static control, the number of cells is viable. You can see it is less. Okay, because the control is static. Okay, and whereas when we keep on increasing the pressure, okay, so with increasing pressure we can see there is a substantial improvement in the number of cells getting adopted to create the focal adhesion points. Okay, with respect to shear stress. Okay, and the same can be validated from the figures which are given below. Okay, regarding the cell viability as well as the OD values. Okay. The story is almost similar with respect to the cell differentiation. Why differentiation we have performed over here is we were working really on the mesenchymal stem cells. So now we know that if we are providing some kind of external forces to the to a premature cell, like a stem cell, then the stem cell will going to have differentiation due to its Q-ness property. Okay, so that's why we just want to confirm that. Okay, so how the differentiation is basically getting sustained in case of shear, say, in presence of shear stress. Okay, so what you can see that in presence of shear stress, the cell differentiation phenomena is almost similar to the static control. Okay, so so with respect to the cell differentiation, the shear stress, or rather the magnitude of the shear stress which we are applying within the microchannel in a linear fashion, it is not really doing any kind of significant changes in the differentiation process. Okay. Okay, so now the main conclusion is like, so what is happening in case of say the temperature means if you are having a regulated temperature and if you do not having a regulated temperature. Okay, so whatever the regulated shear stress we are talking about, so regulated shear stress it comprises in addition to the temperature regulation as well. Okay, so our hypothesis is that why or rather our first question is that in case of regulated shear stress we have found that the cells they are more viable. Okay, so regulated shear stress it comprises of the regulated velocity as well as the regulated temperature if you consider. Okay, what is happening in both the cases, particularly if you talk about say the case 1 or the this figure A. Okay, so in figure A what you can see, in case of static cell culture conditions we are having few number of cytoskeleton which are getting recruited or which are basically helping creation of the focalization points between the matrix, extracellular matrix and the cell, resulting in a say low density signal transduction. Okay, whereas when we are transforming the static condition to a dynamic condition in presence of shear stress, what we can see that more number of cytoskeletons they are getting recruited in order to oppose or prevent the deformation. Okay, and that results in creation of more number of alpha and beta subunits of the focalization points. Okay, resulting in a greater sense of signal transduction from the extracellular membrane to the nucleus. Okay, so thereby it helps in sustaining the cell viability. Now, if we incorporate the temperature parameters into it, okay, so here you can see in figure B. So in figure B, we are having the cells over there, and let's say these are the ice crystal growth due to the cryopreservation phenomena. Okay, and we are having the focal point of adhesions, the oval shape due to this shear stress. Okay, now if we are having the cryopreservation phenomena in a very instantaneous manner, then what will happen? Suddenly, at the extracellular fluid, the fluids will starts transforming into the ice crystals. Okay, so there will be more number of ice crystals between the cell membrane and the extracellular membrane, or we can say the extracellular matrix, or even the substrate of the microchannel. Okay, and that will result in producing hindrance in the formation of the focal adhesion points. Okay, so there will be the result will be that less number of focal adhesion points will be recruited if we are having an instantaneous dip in temperature or instantaneous thermal shock over there. Okay, and simultaneously these crystals, what they will do, these ice crystals, they will be very much vulnerable for the cell membrane to damage them. Okay, and thereby the cell viability is going to decrease rapidly. Now, the next case is that what happens if we bring this ice crystal growth in a very say linear fashion. That is, say we are starting from say 300 Kelvin, okay, and we are go down to 240 Kelvin. Okay, in a linear fashion, in a say at a rate of say 1 Kelvin minus 1 Kelvin per minute, then minus 2 Kelvin per minute, minus 5 Kelvin per minute. Okay, so in that case, what will happen? You can see that there will be development of the ice crystal formation between the cell membrane and the substrate, but that will not be in a say overcrowded fashion. It is going to have a very slow development or slow populations of the ice crystal between the cell membrane and the substrate. Okay, and that results in having a sustainable growth or a sustainable positioning of the focal adhesion points between the cell and the membrane, which will perhaps promote the cell viability, or which helps in preserving the cell viability to some extent. Okay. Now, yes, obviously, when we perform this cryopreservation phenomena, in order to say in order to maintain isotonic condition within the cell, we are providing some cryoprotectant over there. Now when we are providing cryoprotectant in cryopreservation of say type 1, in that case what will happen? The cell, we are exposing it to a say hyper sugar content, okay, which again is not a very healthy conditions for the nucleus of the cell. Okay. Whereas if we are going in a regulated fashion for decreasing the temperature, what will happen? That regulated fashion of decreasing temperature, that can also be used in a supplementary form with having a lower cryoprotectant agents. Okay, so if you are having a say lower concentration of cryoprotectant agent, whereas we are slowly decreasing the temperature to a cryo temperature, in that case we can address two issues. Number one is preventing the cell membrane to get damaged due to the formation of ice crystal. Number two is preventing the cells from getting diluted. Okay, so that is what we are doing over there. Okay, so I think that was my last slide. Okay, so these are some of the ongoing activities in our labs, where we are mostly developing the substrates with the silk fibroin. As we know that silk fibroin is a natural extracellular matrix. Okay, so these are some of the results. I'm not going to that, so just going to the acknowledge. So these are my our lab students who had worked for say for the last 5-6 years in developing or in transforming a lab of a say purely mechanical engineering lab into a interdisciplinary engineering lab, where we have lab members from civil engineering to pharmaceuticals to biotechnology. Okay. So lastly, I want to acknowledge the funding resources from various funding agencies, which help us in developing or transforming the research into from the lab to a certain level of TRL. We are not yet achieved to say went to the market, but yes, that's all. Thank you.
Any questions?
Hi, Arindam. It's a very interesting kind of work. So I believe that you are more working on in an area where we want to see the preserving this mesenchymal cell, and or the effect of the fluid shear stress on determining the
fluid shear stress as well as the
preservation of the mesenchymal cells. So you showed some data where you show that the shear stress or the flow can actually alter the mesenchymal stem cell differentiation.
Differentiation, not really, it is altering. It is we have tried to measure them. You can see here that it is not that much significant.
Means I believe that
it is not altering.
Yeah, I believe that it is quite difficult for only wall shear stress or the flow to determine a cell differentiation state. Yes. Because if you look at every cell in your body is actually in a flow condition, right?
Yes.
You have to take whether it is a blood or interstitial fluid flow, right? Now, if it comes with a combination of factors that actually eventually causes the differentiation to take place, I believe that will be the right model to see how these differentiation stages have been either blocked, facilitated, or it maintained the stemness of the cell. Could have been, interesting to find, I guess.
Yes, absolutely. Actually, in this particular study, here our cells are mostly the adherent cells, and they are arranged in a coplanar fashion.
Right.
Okay, and it is quite early to comment on the cellular aggregation phenomena which was present in a coplanar on the coplanar platform. Now, when we are having, say, the cell-cell contacts, the cell colony densities, as well as the growth factors and different several other cytokines factors, which are collectively imposing the vectors on the colonies of the cells, then only we can, we have seen that there is a change in the differentiation phenomenon as well. But
Right.
that we have not reported here because here the results are of the coplanar structure, not on a, say, spheroids. Okay.
And second question I had is that when you put them, when you're preserving this mesenchymal stem cell and you create a state of flow or the way you have talked about, right? Gradual reduction in the temperature, which we also follow in normal preservation of a cell, right? So do you what is the main reason of
No, so in our lab normally, say, while doing the cryopreservation, so it is like, okay, so we'll be having, say, it at 4 degree, then minus
Then
20,
minus 20,
then
yeah.
minus 80, and
Right.
then in the
it's not like a
the
gradual
cryo camp.
temperature, but it's like a
yes,
yeah.
but there again, so we are going from 4 degree to minus 20.
Right.
Okay, so there is a rapid change
Change
in the temperature. Then minus 20 to minus 80, then minus 80 to minus 196, and we need the minus 196 in that case. But in our case, in our lab, we have developed a cryostage platform where we are not really going to minus 146, minus 196. But we have seen that even if you are going to say minus 60 degrees centigrade, that is sufficient for us to do the cryopreservation, provided we are doing it in a gradual fashion, that is, say, 1 degree, minus 1 degree centigrade per minute, okay, then minus 2 degrees centigrade per minute, minus 5 degrees centigrade per minute. So our temperature shocks are in the resolution of per minute.
But
and per
have you
degree
seen
centigrade.
how long these cells can survive? Like if you follow this pattern, or
In a post incubation period, we have not seen for a, say,
Longer
cryopreservation duration of 3 months or like that. Okay, we have seen up to 7 days.
Thank you.
We have time.
One more question maybe because we'll be short of time.
Is there any other?
Yes, please.
I was curious about the effect of time variation of the shear stress on the
it is,
cells. I will also. What is the value in second minus 1 of the shear stress or shear rate on the surface?
Shear rate on the surface.
Yeah.
In the experimental value we are talking about.
Yes.
I believe we didn't mention here the numerical values we have given over there. No, experimental values also we are having. So here it is mostly, it is very minimal, say 0.002 microbar. Okay. That is the maximum value we are getting, and we have derived, we have given from 0.001 to 0.003. It is a very minimal, because as we are working in the micro channels where the Reynolds number is less than 1. Okay, so that is something critical over there. Since these are some of the early results where the channels which we are considered to be a linear channel, a straight channel. But now we are working in serpentine channels, where we are getting much more stress over there due to the Dean numbers also. So that is something which are not presented over there. Yes, but the shear stress and temperature is different things. Two different independent phenomena. One is the shear stress due to the flow, or the regulation of the flow, and the second one is the regulation of the temperature. So, say in one condition, we are having a fixed flow rate with regulated temperature. Then the second condition, another flow rate with regulated temperature, like that.
No, you mentioned of thermal stress. So,
Yes, sir.
you have any plot for thermal stress?
No, sir, we are not given the plot over there.
This
yes.
is only fluids here,
This is fluids. No.
or combined.
The results are the combined, but the
Combined.
thermal stress, the Nusselt number plots are not given there.
Not Nusselt number, even you have stress. In millibar also you will get same stress, na? But you have not quantified. Okay.
So maybe we can continue.
Okay, you can continue during the lunch time. Running out of time.
Thank you.